So welcome to the 22nd episode of Tokyo Alumni Podcast. Today, our guest graduated the American School in Japan in 2008. Upon graduation, she attended Stanford University, where she majored in human biology with a concentration in neurobiology and behavior. She is currently a PhD candidate at the University of California, San Francisco, in the Department of Bioengineering and Therapeutic Sciences, and is developing CRISPR-based gene therapies for rare genetic disorders. She lives in San Francisco with her husband, Andrew, and 2 dogs. Welcome to the podcast, Serena.
Thank you so much for having me. It's so good to see you.
Yeah, good to see you too. It's been a long time. So
it's been pretty long, huh?
Yeah, people who don't know, you're Serena's brother and I. We graduated together, so we go back quite a ways. So today we'll talk a bit about COVID-19, working at the ground level, actually testing the things they put in the back of your throat. And maybe you can explain to us exactly what's going on there. Talk a bit about the genetics and this CRISPR, the genes-based therapy, how that works, and anything else, I guess, that sort of naturally transpires from those major topics.
Definitely.
So let's start off with just, what is basically going on in the world. I just checked as of this morning, I believe July 3rd, right? It is in the United States, I think it was over 2 million cases, right? And in California, where you are, over 200,000 cases. So can you explain to us what your role has been in regards to...
Absolutely. I guess we should go back to, like March 17th, March 16th. I think that's when San Francisco enacted the shelter-in-place order. And I believe it was one of the first counties to shut down. So San Francisco itself right now in July is reaping the benefits of that early shutdown. But California as a whole, oh dear, we're really seeing that second wave. I guess all I can say is wear a mask. That's really it's really just that simple. But for whatever reason, some people aren't adhering to that. And so that's why we're seeing a lot of huge spikes. So ever since shelter in place back in March, I've been volunteering at the Chan Zuckerberg BioHub, actually conducting the QPCR test to see if those nasopharyngeal swabs that they put down your throat. I call them brain samples because they go so far back. But I'm one of the volunteer scientists who's actually taking those nasal swabs and then doing these experiments to see if there's live virus in that sample.
So when you analyze the samples, what exactly transpires? I just had mine actually 2 days ago, and I just got my results negative. But I just have no idea what's going on. They just stuck it in my nose, and then they went away, and they just told me I don't have COVID. So I mean, what is sort of like the 1 minute version of what exactly are they looking for?
That's a great question. And so if you think about it, you get your nasal swab, and then they put that nasal swab inside of a tube. And that tube has this liquid that preserves all the RNA, all the genetic information that's collected on that swab. And then they get packaged up and sent over to a lab. And then let's say that I receive these thousands of tubes at the beginning of my shift. And we essentially use these robots to help transfer the liquid contents of those tubes into this format that we can load onto a different robot where we extract out the viral RNA samples from the nasal swabs, and then we can quantitatively detect if we see any live virus in those swabs. So it's a really, really accurate, really precise method to see if you carry the coronavirus.
That's interesting. And, I've read a bit about the antibody tests not being as accurate. So how do they compare? And also the COVID-19, tests where you do get the nasal swab or the brain. How accurate is that? And I mean, are there ever times where it's not accurate?
Yeah, I would say the antibody tests, I don't know too much about the antibody tests. It's true that they are not as accurate as the qPCR test, which is the RNA-based test. The main difference, though, is that the antibody tests will essentially tell you if you've ever been exposed to the virus. It'll tell you if your immune system has ever mounted a response to the coronavirus, and then you can track the remnants of that immune response. So that's less sensitive, but in my opinion, it's more important for us to determine if you're walking around with the live coronavirus, if your body is incubating this virus and then passing it along to others, especially because we know, especially for people perhaps in our age range, we can be really asymptomatic. So those qPCR-based tests are actually really, really sensitive. I don't have the exact numbers off the top of my head, but you can think about it as if you have like a kiddie pool, you know, one of those swimming pools filled with water, and you drop in, you know, one pebble into that kiddie pool. The qPCR test would be able to find that one pebble amidst this big pool. So it's a very classic, very well understood method of detecting RNA. So I feel very confident in the level of accuracy and sensitivity. But of course, with any test, there's variation, especially at the point of actually swabbing. When you swab your nose sample, you know, it could be that if you're at the earlier side of infectivity, maybe they don't touch the part of the sputum that carries the virus or something like that. So there's always like a little bit of variation, but I would say if we could all try to test ourselves more often, I think that we would be in a much, much better place. Yeah, more testing and wear a mask. And I think next year we'll be looking a lot more normal.
Yeah. I hope 2021.
Yeah.
It has to be better than 2022. But I guess I don't. I shouldn't jinx it.
I know, right?
But with this COVID testing, I know we don't want to get too much in the field of speculation, especially, you know, because you're a scientist. But at least within what you know, the cost of COVID tests, I understand, is getting cheaper because, you know, more people are doing it. Is the direction that, you know, if we do continue to see an abundance of COVID testing, even in 2021, are we looking at COVID testing being, you know, cheap enough so that people can be doing it very frequently, like once a month?
Yeah. I don't really work on the financial end of the testing, which I know is a big deal, but I will say this: at the Chan-Zuckerberg BioHub, where I'm doing these tests, we provide all of the testing for free. So we've processed and sampled well over 50,000 samples, all for free. We do over 1,000 tests a day for free. So it is doable. That being said, we do have an endowment for this testing facility. But I believe that a lot of the biotech companies that provide the reagents for these tests are being very generous and donating a lot of material. So I don't believe that the facility that I'm working at is the only place. You know, I don't think that we're the anomaly where we can provide free testing. I think that it's very reasonable that moving forward, as people need more testing, as the supplies become easier to get, I think that we should be able to lower the cost. Or I think that is, as a scientist, within our field, that is our ethical responsibility to do that.
Yeah, that's incredibly intriguing. That's how generous some of these groups have been, and hopefully it'll continue on.
That being said, we do have an army of volunteer graduate students who are really, you know, actually doing the tests. So that's something to keep in mind. You know, the people who usually are doing the tests aren't graduate students, so they require obviously a salary and things like that. But this model, I think, of using life science graduate students to be a part of this testing pipeline is just brilliant. And there's graduate students everywhere who can help with this. So at least in California, Gavin Newsom, I believe, I don't know if it's just a California mandate, but anyways, they essentially allowed academic research labs to start processing patient samples. And that was a game changer, which is what allowed graduate students like myself to go in and volunteer with real patient samples. So if that mandate can be passed along to other states, perhaps it already is, maybe this model could really help in the future.
Yeah, it was off air. You mentioned that this was just a fascinating period where in history where graduate students were in the front lines, right, analyzing these tests. So you also mentioned off air a bit how you were working on something prior, and would it be correct to assume that a lot of other people who were working on other medical related, PhDs, it's sort of all hands on deck and everyone is just COVID-19 defense mode right now?
I think that's really, that's pretty much the case as what happened. So at least at UCSF, they shut down our research labs. I guess shelter in place started March 16th. We were not allowed to go back to lab on March 18th. So that was really, really devastating for our research progress. I'm working on a PhD thesis that requires like long-term experiments and things like that. So I couldn't really go in and do my experiments anymore. But instead, I got an email that was circulated to a lot of the biological sciences PhD students to come in and be qPCR ninjas for the COVID tests. And I'll tell you this much, so many people signed up to do this. It was it was just like an all hands on deck situation. And even if some students, didn't sign up to do the actual qPCR test, but rather they shifted their research focus and instead looked into more about the biological processes of the coronavirus itself to help come up with, different docking mechanisms to treat coronavirus or do sequencing for the mutagenesis rates of the coronavirus as it spreads throughout population. So it was really amazing to see how a lot of even though a lot of PhD research and postdoctoral research was really, really devastated by this closure, we all kind of just like pivoted and did what we could and tried to help the community.
Wow. So like on one hand, it's like, I'm so happy about the valiant efforts of the people in your community, but it almost like, I don't want to focus on the negatives, but it just angers me that much more that there's people who can't even wear a mask when you have these other people who are like volunteering, their precious time and hours.
Yeah, man. Well, aside from the fact, and I'll say this much, as a scientist, yes, I'm technically on the front line, but I'm in a laboratory. I'm dealing with the patient samples, but I'm not standing there amongst, I'm not on the nursing floors or like the emergency rooms or ICU floors. I mean, that's really who I'm thinking of here too. But I want to say something about the masks. I've been so furious about why people don't adhere to wearing masks. It's so simple, especially when I think back to summer day camp. Did you ever do summer day camp?
Yes.
Yeah. And, we're with these kids. It's the middle of summer in Japan. It's so hot, so humid. And I remember that some of our kids would be wearing masks all day long because they had a little bit of a cough. Mm-hmm. And if a child can do that in humid Tokyo summers, an adult here can absolutely wear a mask. So that's my little rant about masks. I'm in shock that someone could put their minor discomfort over other people's lives. I don't understand.
Yeah, I absolutely concur. It's quite irritating.
Yeah, absolutely.
So, we've spoken about COVID a bit, and I want to sort of switch gears into this genetic testing you do. It's something I find incredibly interesting, especially being a psychology teacher, right? This idea of approaching the issues at a genetic base, right? Because so much of who we are is already based on our systems, yet there's this study about epigenetics, right? And how people's genes themselves can be molded. So it's kind of like this very dualistic thing where on one hand, we're born a certain way. Like I'm 5'8". I tell people I'm 5'9", but that's mostly genetics. But in a certain way, depending on, how I was raised, those genes could change. So maybe, can you tell us a bit about what you were doing at least prior to all this COVID madness?
Sure.
What your studies encompass.
Yeah, absolutely. So in my doctoral research, I'm developing a gene therapy to rescue one of the most significant genetic forms of autism. So what that means is that's kind of like a big sentence there, but essentially I'm working at the level of the DNA. So with the development of like next generation sequencing methods and also the development of the human genome, we can really study genetics, and we can tie that genetics to, you know, when things mutate and then really devastating diseases happen. And so I work with this tool called CRISPR, which is a really, really precise gene editing tool, where we call it sort of a genetic scalpel, where you can literally go into a very specific region in your genome and then edit that mutation. So that's pretty much what I'm doing in my research, but specifically targeting a gene that's related to autism when it gets mutated.
Interesting. And is that process, so when you're figuring out, you know, the mutation of the genes, sort of for the normal person, because I think already at this point, you know, I'm a chimpanzee.
Yeah.
I don't really,
Stop.
I'm starting to get confused of how is it actually done though? I mean,
you know, I'm standing on the backs of giants who have produced so much literature, so much research over this last many decades. So I was really fortunate where I was able to go in with a little bit more information and try to fix the problem. Let me back up a little bit, because a lot of people don't realize that autism spectrum disorder has a genetic component to it. You know, the autism spectrum disorder is one of the most common neurodevelopmental disorders in the entire world. But it's tricky because to diagnose autism, it's all clinically based off of human behavior, really complex human behaviors. So what a number of researchers did a few years ago was they took patient samples, like blood samples or even saliva samples of individuals with autism spectrum disorder. And they also took samples from their parent who did not have autism. And then you can sequence the DNA of these cohorts of individuals and their entire families to see what changed from the parent to the child. Did anything mutate? What happened? And what we found is that over 100 genes in the human genome randomly mutate, lead to a development of autism. So I'm specifically targeting one of the most commonly mutated genes that's known to cause this disorder, which is, it's a little nitty-gritty, but it's a sodium channel in our brains. So the way that our neurons communicate to each other get disrupted when you have this mutation.
that's fascinating. So when it comes to the genes mutating and, you know, you're comparing the parents to the children, you know, at least based on high school biology, that's the limit of my knowledge. I remember learning about, you know, how children with Down syndrome have it's very clear cut, right, that they're missing a chromosome. So when it comes to autism, how clear cut is it that, like,
oh my gosh, that's a really, really good question. It's difficult to sequence these mutations, or at least to identify these mutations. It requires, you know, thousands and thousands of these family models to really be able to parse apart the differences, because from passing from parent to child, inevitably, you know, our replication machinery for DNA replication changes a little bit, but with enough sample size, you can detect even the most minor base pair change. It's magnificent. And I didn't do that in my research. Someone else did that already, so I'm really thankful for that. But it's not as clear cut as what we learned in the sequencing method.
So we have this data that you've extrapolated upon, like, about, you know, the parents' genes and children's genes. So then how is this all being utilized? So what is sort of the end all goal of getting this information?
Yeah, a lot of people are using this amazing, rich data set for many different reasons. I'm on the therapeutic development side, so my goal is to develop a gene therapy to actually fix this mutation. So in live humans, that's my goal, is to develop a true treatment as opposed to a lifelong medication that someone might have to take for the rest of their lives. I'm developing a viral-based treatment that could actually go in and truly change the framework of your DNA, so you would not need medication anymore. That being said, I work on in the extremely early side of everything in terms of the research process. So I work with stem cells, and I work with mice. So who knows, what will happen with research? But even if I can't make my goal of developing that treatment, I think whatever I am able to produce will still contribute to this, to the field. So I'm on the developing the treatment side, but some other people are on why do these mutations happen? What causes those mutations? That's really, really important. I don't know the answer. It could be, environmental toxins. It could be, who knows what it is on that front as well. The brain is amazing, and it truly is a frontier in terms of the research world. So things change, things develop really, really quickly. So I'm just trying to hold on and contribute what I can.
Yeah, that's incredibly interesting how you mentioned it, I think, off air as well, the whole thing about experimental areas, right? So you're always sort of in the front end of the most cutting edge ideas in regards to, how to make the world a better place. And so specifically, you mentioned autism. When it comes to this, sequencing of genetics and looking at patterns, are there other areas apart from autism that you guys focus on?
Oh yeah, absolutely. My lab studies a lot of different disorders. Some aren't even studying specific disorders. They're just interested in like the development of our chromatin and how they package into a cell's nucleus. Another disorder that we're targeting, or actually we published this data a few years ago in Science. We were essentially able to use a CRISPR-based method to treat one of the most significant mutations that causes obesity. So for that, we used mouse models where we were able to take these like really, really obese mice. They were probably the size of like 3 times the weight of a typical mouse body weight, and we were able to do this DNA repair on those mice, and then they developed into typical body weight. So that was kind of interesting, and it was really drastic to see this change from truly morbidly obese to healthy, functioning, and fully developed mouse.
So when you say genetic obesity, is that kind of like with humans, would that be something like a thyroid issue? Are we talking about something much more severe?
It's kind of similar. Yeah, there's so the gene that we went after was called SIM1, and it's more involved in your pituitary glands. So it absolutely is part of your signaling pathway for like leptin and ghrelin, which is like the hormones that's produced when you're hungry or when you're satiated. It kind of regulates our intake of food or energy. So it's amazing. I love science because it's like before I really got into research, I never knew that something as intangible as like food regulation could be tied to something biological and even genetic.
It's interesting. And then you were involved with this, concept of biology and human development as early as your undergraduate at Stanford. So was there sort of a catalyst year where you were like, this is it, this is what I'm doing?
Yeah, absolutely. If anyone knew me at ASIJ, I was never really the science, the humanities, the humanities side of the high school. But when I went to Stanford, I was really exposed to research. It really just blew my mind. I was really motivated to learn more about biology when I learned about psychiatric disorders, just because it really frustrated me that there's no movement in terms of safe and effective medications for a number of different psychiatric disorders, and rather these individuals are put on lifelong tranquilizers or things that are just masking the symptoms rather than actually treating the the genetic, or not even the genetic, but the biology itself. So that was my main motivation to go into research, was to develop treatments for untreatable diseases. And I was really lucky because I had a work study opportunity to work in a research lab. And so I started as just washing glassware, and I was washing dishes. But the professor that I worked for eventually started giving me, more research responsibilities. I did a lot of monkey behavior studies back then, and then I just sort of built from there.
And now this is all during undergrad.
Yeah, this was all during undergrad. Yeah.
Wow.
Yeah. So I really had back in 2008, when I graduated from ASIJ, I had no idea that I would be going down the PhD track. I still, it still shocks a lot of people, but to me, it all kind of makes sense. I've always been motivated to help others, and I think science is a really cool way to do that.
Yeah, it's really interesting to hear about all this, how, as you said, it connects to everything, right? Whether it's obesity, autism, a lot of our cognitive functions. I don't mean to sort of get too philosophical, but a question I often face, just as, a psychology teacher, so obviously we do something far more simplistic than what you do, but I think the question still resonates, is nature versus nurture. Right? The idea of our genes versus, how we're developed. So now that you're so, deep into this study of genetics as well as epigenetics, what has been maybe your greatest findings in regards to how much our genes control us, and to a certain degree, how free am I from my genes? Like I know I'm me with my genes from my mother and father, but how much can I change?
I think about this a lot when I'm thinking about organoid models. So organoid models are essentially 3D or like miniature brains, essentially, where we're trying to model this cell type and then this cell type and this, and then to put it all together and then test different drugs on it, right? We're trying to grow miniature brains, essentially. And we're able to get, the genetic markers down. We're able to get the layers of the cortical, the cortical development down. But is that truly a brain? No. Genetics is not enough to dictate human behavior, but it is the blueprint. It's the launching pad for it. What am I trying to say? I think that your genetics dictates a lot of what you might be, but not who you might be, if that makes any sense.
Yeah, I see what you're saying. And I'm just wondering, so when you look at these case studies, and I'm sure you've seen thousands, what would be the biggest deviations you've seen where like someone, should have been a certain way based on their genetics, but they turned out the other way? And have there been vice versa, where like someone clearly was genetically meant to be a certain way, but their lifestyle and whatnot changed who they became?
Oh gosh. I do know that, especially in terms of stress response, especially for developing fetuses, like that is huge. Intergenerational trauma is definitely something that I'm really interested in, and how trauma in one generation passes to another. And some people say that you can see that in the epigenetic factors of like methylation. And I think that's fascinating. I probably don't know enough to really give a satisfying response to this, but I do feel that even though I studied the genetics of life, that's never going to fully encompass what life really is. So I'm very, I'm humbled by what we can do in a research lab, but human life, especially human development, is so complex, and it's so interdependent with other influences in life. But hopefully, as we learn more about genetics, and hopefully as we get better about modeling these things, maybe we can learn a little bit more.
So we're going to switch gears now, going from COVID to genetics to something totally different. But just as well, I'm not going to say just as important. It's obviously subjective, but you have an interesting background of being this Asian American and you were in California till age, I guess, right before teenage years. Spent most of your teenage years Japan, and then now you're back in California. Overall, what was the experience like living in Japan, being Asian American, and then going back to the States? Like, did you feel a reverse culture shock or culture shock? I don't know, it's all semantics, but yeah.
Oh, yeah. So interesting. So I'm a fourth generation Japanese American. I was born in Berkeley, California, and I didn't speak a lick of Japanese when I moved to Japan. So I moved when I was in sixth grade, so I guess I was 10 or 11. And so up until that point, I had a very, very typical middle-class Japanese American lifestyle. I lived in the Bay Area. I went to public school. I went to a Buddhist church where I congregated with other Japanese American families, and no one there spoke Japanese either, but we all really liked Japanese food. So I was around the culture, but I was never really, I didn't really identify as Japanese truly, just because I felt more of a Japanese American, I guess. Mm-hmm. So when I moved to Japan, oh gosh, that was a really hard transition for me, largely because obviously I'm 100% Japanese. I look Japanese, but I don't speak the language. I don't adhere to a lot of the societal expectations of what a Japanese woman might behave or speak like. And especially when I was at ASIJ, I feel like a lot of people didn't understand that you could be American and not white. So everyone assumed that I was half white, half Japanese. So that was really challenging for me when I got to ASIJ, where I had to defend my Americanness, and had to give a little bit of, and then when I left Japan, I loved my time in Japan, and by the time I graduated, I was very proud, but I got a 5 on my AP Japanese test. So
yeah,
big deal.
That's a big deal, yeah. Especially
big deal. Mrs. Virgil really got me there, but. When I moved back from Japan, now going to Stanford, back in California, then I became the international student. It was so funny because here I was in Japan saying like, I'm truly American. And now I'm in America and I've been dubbed as the international student from Japan. So people would be, man, you speak really great English. And I feel like, yeah, that's pretty much the only language I can speak. But I've really come to terms with the fact that a lot of people just aren't familiar with, this migration pattern. And I take it as an opportunity to kind of open minds a little bit. But yeah, it was definitely a flip-flop, I guess.
Yeah, that's fascinating to hear. So you're sort of like a foreigner in both cultures.
Exactly.
Sort of part of this tribe, right? It reminds me of when I actually just flew back to Japan 2 days ago. They were putting together all the people who were Japanese, and they got to go first. And I had my Japanese passport, and this person kept stopping me, and I kept speaking to them in Japanese, saying I'm Japanese, and they kept stopping.
And you speak fluent Japanese.
They were just convinced, and then they kept saying DOD, Department of Defense. I said no, I'm not in the military. I wouldn't survive a day in the military. And then I had to get my passport out because I originally I was just thinking, come on, just like let me.
Yeah.
But then I showed her, and then I kind of had to apologize to her. Very, very Japanese of me, right? I was apologizing to her, saying like it's okay, like I know I don't look Japanese.
It's confusing. It's really not that confusing, but it's so funny. I also find myself after living in Japan for so long, I've definitely incorporated some of the humility of, living in Japan and always apologizing first. Oh my gosh. And being so damn accommodating to everyone.
Yeah. And maybe that's why things like the mask really rub us the wrong way. Right?
Definitely. I think you're absolutely right. It grates against my core values, which were probably developed in Japan.
So in that sense, it's cool that it's like, as I've started this podcast, it's sort of like we're all part of this tribe, right? Whether or not we live in the US or, God, I mean, just in the first 22 episodes, I think we've had people living in Peru. I can't remember. There's at least 6 countries. I should remember, but there was at least
That's awesome.
6, 7 countries, but we're all sort of from that same tribe.
Yeah.
So as we wind down this podcast, at the very end, I like to have the guest speak for, a minute or two. Some people go 5. You don't have to go 5, but if you want a few minutes about, what's coming up in their lives for the next few years, next few decades, what's to come. So please take it away, Serena.
Sure. I guess the biggest thing that I'm going to try to achieve in the near future is to graduate. I really hope that I can graduate in the next maybe year or 2. This shelter-in-place order really set my research back by a good chunk. So I'm trying to get things back online and continue to hopefully one day publish a paper where I can show that I was able to develop some sort of a therapy for that genetic form of autism that we talked about earlier. That's my biggest goal. After I graduate, I really hope to kind of pivot and go more into the biotech investment side, or perhaps more of an entrepreneurial side. There's so much exciting, really innovative work that's being done in the Bay Area, kind of melding together technology and basic life sciences. So I hope that I can find my way into that arena somehow. But I imagine that I'll stay near the Bay Area. It's where my husband and I are both originally from, and we like the weather out here. But something that I'm also really passionate about is trying to show others that might not look like a typical scientist, that science is for them too. So I'm really interested in, I'm very evangelical, I would say, about making sure that, especially children in the Bay Area or around the world can really see themselves as scientists. Because I definitely didn't think of myself as a scientist. I still don't really think about it because, all the scientists that I was ever exposed to when I was younger were, kind of crazy-haired white guys. So,
Yeah.
So that's something that I'm trying to really incorporate into my life at UCSF by, hosting more volunteer opportunities, going out into the community more, and doing more outreach. So I hope that no matter where my life takes me, I hope that I can continue that journey.
Well, thank you for being on this episode. We appreciate all the work you're doing with the COVID-19. And hopefully next year things will be a bit more normal. Yeah, it's been nice talking to you.
Thank you so much for having me.
Yeah, and for maybe the 7 people who are listening, wear a mask. Please
wear a mask.
Just do it. Just do it.
Awesome.
On that note, thank you again, and see you around. Goodbye.
See you around.